express my gratitude to Andrea for accepting my invitation to present and share her work with us today. So thank you, Andrea. Dr. Gaedigk played instrumental role in numerous studies performed over the last 25 years in the field of pharmacogenetics. And I had the privilege actually to work in her lab for almost 1.5 years while I was a student, a PhD student at UMKC. In her lab, it was my first experience or exposure to the field of genetics, and my work there with Andrea, Roger, and everyone in the lab actually inspired me to shift my interest toward the field of genetics. So she also helped me during my transition from UMKC to UT. So thank you, and Roger, for all the support, Andrea. So now just a few or brief introduction about Dr. Gaedigk. Dr. Gaedigk, she received her master's and PhD degrees in biology from the University of Stuttgart, Germany. She got then her training as a postdoc fellow at the Hospital for Sick Children in the Department of Clinical Pharmacology in Toronto, Canada. Then in 1996, Dr. Gaedigk moved to Kansas City, Missouri, where she accepted a position as Associate Director in the Pediatric Pharmacology Research Unit Lab at Children's Mercy Hospital. She also joined the faculty as Assistant Professor at UMKC School of Medicine, and she then promoted to Associate Professor in 2005 and to a Professor in 2014. Dr. Gaedigk also holds an adjunct faculty position at UMKC School of Pharmacy and the Department of Clinical Laboratory Sciences at University of Kansas Medical Center. Dr. Gaedigk has long-standing interest in pharmacogenes. She has extensively worked with cytochrome P450, more specifically CYP2D6, and has published over 180 peer-reviewed articles in national and international journals. She presented also more than 190 abstracts, 3 book chapters, and reviewed over 300 manuscripts. Dr. Gaedigk, she's a member of the editorial boards of different journals. She's also has been actively involved in many societies like American Society of Clinical Pharmacology, ASPID, the International Society for Study of Xenobiotics, and the American Society for Human Genetics, and a founding member of the new Pharmacogenomic Research Network. She's closely also working with the Pharmacogenomics Knowledge Base and the Clinical Pharmacogenetics Implementation Consortium, the CPIC. So I'm sure we're still missing many other accomplishments and positions, but just for the sake of time, thank you again for sharing your and presenting your work with us today. And yeah, it's all yours. Thanks.
Thank you, Noor, for this very kind introduction. So the pleasure is all mine to present to you today what I'm up to over the past few years. And so my focus or my time has really been allocated to launching the Pharmacogene Variation Consortium. And as Noor mentioned, also work closely together with some other PGX resources. So this is what I'm going to present to you today. So my objectives are that you understand that people metabolize and respond to drugs differently, understand that many drug metabolizing enzymes are highly polymorphic, and also learn about pharmacogenetic resources. So we will not be able during the talk to get too detailed with all those, but hopefully I will spark your interest and give you the resources so that you know where you can go to then at a later time on your own. So I want to start with a quote that was made over 100 years ago, and which is really still valid today as it was back then. And it says that variability is the law of life, and as no two faces are the same, so no two bodies are alike, and no two individuals react alike and behave alike under the abnormal conditions, which we know as disease. And so this, of course, also applies to pharmacogenetics, which is really the genetic contribution to drug metabolism and response. Now, the only difference is that while there is a lot of phenotypes are visible, like skin color, hair color, eye color, height, and so on and so forth, we often don't really know how are we different when it comes to drug metabolism and response, because you only exhibit a phenotype for that once you are exposed to the drug. And so how are we different? Age, gender, ethnicity or ancestry, body composition, which means weight. There is really a lot of work forthcoming about obesity, how that contributes to so many different things, including drug metabolism. Then, of course, the diet, the environment, your history, illnesses, and of course, your genetic makeup. And all these different factors affect the way we respond to medications. So what is important to really consider? So how does the body process any drugs? So first of all, the drug needs to be absorbed. Typically, drugs are given orally, so you swallow them. Then they get into your digestive system. From there, they need to be absorbed. They need to be distributed, which is usually happening through the blood. So they need to get from your digestive system out into circulation. Then drug metabolism, a lot of that happens in the liver. So it needs to be taken up from the blood into the liver. And then, of course, excretion is also extremely important, that we get rid of metabolites or the drugs themselves. So not every drug, of course, is metabolized. And some drugs are also metabolized into rather toxic metabolites. And it's also important to realize that some drugs are actually prodrugs. So they need to first undergo bioactivation so that we do have these active metabolites which can then act. And of course, then the drugs also need to get to the place of action. So they need to find, for example, a receptor. Certain drugs need to get into your brain where they need to act. So there is a lot going on. And the concept of absorption, distribution, metabolism, and excretion is often also referred to as ADME. So now when we talk about, how people have been treated in the past and are often still treated today, is that you do have a patient group, and all these people are given the same standard prescription and the same standard dose. But some of these might have a normal drug response. Normal meaning as expected, because they probably fit within that average person on which the drug was developed. There might be people who have some response, but not as good as expected. Some people have, an adverse response. They might have side effects. There might be people who really have a toxic response, where there could be very severe side effects or adverse reactions. And then there might be people without a response. And so what we understand or often call precision medicine or personalized drug therapy, the idea really is that what if we could use genetic analysis, if we would know what kind of genetic variation contributes to these different responses, if we test people up front, and then if we have that patient population, could triage them into responders or non-responders up front, then we could give these people individualized treatment, which would be, for example, choosing a different drug if there is one, or adjusting the dose, or start the dose somewhere different, higher or lower. We could also monitor them more closely to really watch out for side effects. And so what we understand under pharmacogenetics is really the identification and recognition of variants in a number of single genes that have a clinically relevant effect on the kinetics and often also really the dynamics of a particular medication. Now, as you see it here in the graph, where we have green people and red people, there are of course also people who are, really kind of in between, like those who do not respond as well, or those with no response. And so the picture is really not as clear cut yet. And so pharmacogenetics is definitely not the silver bullet. It does not answer all the questions, but it is really one piece in the puzzle to improve therapy. There are different pieces that need to come together, and as I indicated before, age, history, and so on, and so forth, all these play their role, and pharmacogenetics is just another piece in the treatment that can really, under certain circumstances, illnesses, potential drugs to be given, play an important role to just improve therapy and response right from the get-go. Often these days, there's also a lot of pharmacogenetic testing not done up front, but only after a person, for example, has experienced adverse drug reactions. So once a person has gone through a number of drugs and there was no response, or if a person has had bad reactions, then pharmacogenetic testing is consulted to find answers. And so there are different models of practice in different institutions, but there's also so often pharmacogenetics is not even part of a treatment plan. So there are a number of drug metabolizing enzymes with documented clinical consequences, and I'm just listing some of those here with some drugs. So warfarin is an anticoagulant that is often taken, especially in older people. It's a blood thinner. And so part of the variability among the people is in a cytochrome P450, especially 2C9, and VCOR, that is a dynamic, a pharmacodynamic gene, which has to do with vitamin D levels. So it's these two together that explain a part of the response to warfarin. And a blood thinner, it's easy to imagine that if the blood becomes too thin, and especially if you have older people, if they fall and there's a bleeding episode, that could really have a fatal outcome. So bleeding is a big concern for warfarin, and genotyping guided prescribing, especially for dosage, is now implemented in quite a few institutions around the country. Another example is CYP2C19, which is important for omeprazole, which is a proton pump inhibitor, and clopidogrel, which prevents platelets to stick together. So that is an important drug for a lot of heart conditions. And then CYP2D6 is yet another one, and this is in red because it is one of my favorite genes to work with. And this enzyme metabolizes a whole lot of different drugs, among them tricyclic antidepressants, SSRIs, but also some opioids as codeine. And codeine is actually one of those drugs that require bioactivation. So that on its own is not active, so you don't get the pain relief through the metabolite, which is a morphine derivative. So you need to have some activity of 2D6 to get the intended effect here. And then there are others as well. And so here you can see a selection of drugs metabolized by 2D6. And as you can see here, They come from really different classes: antiarrhythmics, beta blockers, a lot of antidepressants and antipsychotics. And so it is not surprising that this gene has received a lot of attention over past decades, and is also one that is most extensively genotyped these days if a pharmacogenetic test is obtained. And this is also metabolizing dextromethorphan. And so as we go into flu season or cough season, cough syrup, you might or might not know, contains dextromethorphan, and that is metabolized by CYP2D6. So also considering all these different drugs, it is highly likely that, you know, every person encounters a drug at some point in your life that is metabolized by 2D6. And so just knowing where on the spectrum of activity you are can really help to make smarter choices of therapy. And so when we look at 2D6 activity on a population basis, what you see here on the left is a typical activity spectrum as you see in a wide US population. So this graph is from a section of individuals we have done in Kansas City many years ago. And so what you can see here on the extremes, there are individuals who are poor metabolizers who have no activity. On the other end, you have ultra rapid metabolizers. And then in the middle, the bulk of the people, about 80% or so, are normal metabolizers. And then there are also some, which you can see here as a kind of shoulder, are intermediate metabolizers. And so this population distribution looks different in different populations. So in Asians, East Asians, there are less poor metabolizers. There are many more intermediate metabolizers, and other populations are just somewhere in between. And it depends on your genetic makeup, what kind of variants occur in these different populations of what this phenotypic histogram looks like. When you look here on the right, so when what is the consequence of being an ultra-rapid, normal, intermediate, or poor metabolizer? And that is just indicated here in the graphs below. So if you are a poor metabolizer, you might have a much, much higher exposure to the drug compared to a normal metabolizer. And that can lead or cause adverse events. Then as intermediate metabolizers, you are still possibly at a higher exposure. And so it will depend really on the therapeutic window of a drug, whether there is a risk of a particular drug to experience side effects. And then of course, if you're an ultra rapid metabolizer and you are really, really very fastly metabolizing the drug, you might never actually have the level of exposure that is necessary for the drug to work. So ultra-rapid metabolizers sometimes need significant higher doses of the drug to actually work. And so, of course, if you would not have any information about that patient, where on the spectrum he or she is, and you just get the average dose, you might be overexposed or underexposed, especially in these individuals on these extreme phenotype sites. So CYP2D6 is also a highly complex and a highly polymorphic gene locus. And I heard that you have looked at the paper yesterday. So that gene focus paper I have circulated, there is a graph in there which gives you an idea of really the complex structure. And we can look at that later on if there are any questions. But essentially, whatever genetic variation there is, you can possibly find that in CYP2D6. So there are many single nucleotide polymorphisms or SNPs. there are small deletions or insertions, just a few nucleotides, which we refer to as indels. There is copy number variation that can lead to deletion of the entire gene, or gene duplications, multiplications. And then there are also gene arrangements which lead to hypergenes between 2D6 and its pseudogene 2D7, and also unequal duplications where you have multiple gene copies, but they are not identical gene copies, but are really different kind of copies sitting there in tandem. And of course, all the analysis of that locus is complicated by having the two pseudogenes, 2D7 and 2D8 also there. So that makes it really tough from a technical perspective to do genotyping. And we are not having time during the talk to get into really all these intricacies of 2D6 genetic variation. Okay, I will just stop here and ask whether there are any kind of questions so far.
Andrea, just regarding having ultra-rapid metabolizer instead of rapid metabolizer, I remember it was rapid and then they changed into ultra-rapid. I mean, just like basic question, why it's like ultra compared to rapid?
Well, ultra-rapid is a term that has been around for a long time. And actually, depending on the gene, for some genes, there are both of these phenotypes are used. For 2C19, they are using rapid metabolizer and ultra-rapid metabolizer. So certain genotypes are translated into one or the other, depending on what you have. For 2D6, there is no rapid metabolizer group right now. There are only normal and ultra-rapid metabolizers. And the reason for that is that it isn't really fully clear whether there is really, whether it's clinically necessary to pull them apart. Or in other words, they always ask the question, would rapid or ultra-rapid metabolizers require the same or different clinical recommendations? And if the recommendation would be the same, then they try to keep it as simple as possible. Okay, if there are no other questions, I will then just go ahead and start explaining a little bit about nomenclature. So I need to take you back about 25 years, and it's probably some of you here on the other end of the computer might have not even been born then, or were, you know, still really young. So it's a long time ago that a group of experts came together to really think about how can we organize this? How can we systematically capture all the variation that was essentially kind of exploding after there was a lot of technical advance in the late 80s and early 90s. So this was the time sequencing became much easier to do with a higher throughput, although it's nothing of what we can do today. Also, PCR became available and became more robust, and more methods were published. And with all these advancements in methodology is when these genes were more and more characterized in more people, and all these variations were discovered. And so there was really the need to find a system and to encourage the scientists worldwide to really speak the same language. And this is why that star nomenclature was sort of invented back then, and really embraced by that pharmacogenetic community that also kind of formed in the late 80s, early 90s, when all that became available. And the first paper on which that star nomenclature was published was on CYP2D6. So the interest of this gene really goes back quite a long time. And then just about 4 or 5 years later, the star nomenclature was expanded to all other cytochrome P450s because it was really a big hit. And it also became clear at the time that they could no longer really keep track of that on paper or via manuscripts. But then we also had the internet, and this cytochrome P450 allele nomenclature page went up on the web. It was housed at the Karolinska Institute in Stockholm, Sweden, since 2002. And so you could now go online and see what all these different star designations or variants were. And so that really became the go-to resource for variation for these cytochrome P450s. And so this is, oops, that was one that actually a slide I kind of took out. It's essentially the same thing, but you see this is the person who housed it and was leading this page for about 15 years. And then he decided now back in 2015-16 that he really wanted to go on to some other adventures, and this is when the Pharmacogenetic Variation Consortium was born. So I took leadership of that, and also really tried to lift this database as it was before really into the 21st century. So the goal really was to develop a database that is much more interactive, much more systematic. Also, how we define all these alleles needed to be much more transparent. We needed much better described processes in place. And so in 2017, this old nomenclature database was moved to PharmVar. We also work very closely together with the PharmGKB, and this is really in the spirit of working together and try to standardize as much as we possibly can among these different pharmacogenetic resources, because it is really, really hard if we are not using the same nomenclature, saying the same thing. And so I probably also really now need to explain what that star nomenclature is. And so what you can see here on the left is the gene symbol. So the gene symbol, so this is issued by UGO. So there is a symbol gene name consortium. So CYP stands for cytochrome P450, and it's in capital letters because we are doing human genes here. Then the number here indicates the family. So cytochrome P450 is a superfamily of enzymes, and there is a family 1, a family 2, 3, 4, and these all are enzymes that are involved in drug metabolism, and CYPs with higher numbers tend to be involved with cholesterol and hormone metabolism. So this here is the family, and then there is a subfamily, and so this is where the letter comes into play. And then there is the isoform. So when we are on human CYP2D, it's the number 6. So Noor will remember we have worked on macacos, and it was number 17, right? And so different species have different numbers here. So this is the isoform. And then the allele, what is denoted as a star, this is essentially the haplotype variant you see in that individual. And haplotype is important for these cytochrome P450s because, as I will show you, there can be different combinations of all these different SNPs. And so it is important to know which of these SNPs or variants are on the same allelic variant or on the same chromosome. And so this here is how it's structured. And for these resources, so PharmVar now holds mostly CYPs, but we are also expanding to other clinically actionable pharmacogenes. And so we have introduced NUT15 and DPYD. We are also expanding to other genes. We will soon start to work on our first drug transporters. There are also other web-based sites. There's one for TPMT, UGT, NAT, and ALDH. And so for the NATs, we will probably incorporate those into PharmVar also sometimes in the future. So we have started working with the NAT group here. And then for some other genes, there are really just recommendations where people use star nomenclature very loosely. So they are just self-assigning, and that can also really pose a problem. And so for the CYPs, and this was really the beauty of that webpage, that people adhered, at least for those genes, to only using star nomenclature for the most part, if it was officially issued by that site. And so we really encourage people not to self-assign star numbers for any CYPs, but go through us so we can review those, that we can put them on the web page and officially issue a star number. Of course, we cannot force people to do that, so everybody can also publish novel variants they find, absolutely, but in that case, they should not use any self-assigned star number. And it's also really important to realize that these star numbers need to be regulated somehow, especially as we move into clinical application. And I will show you where that plays a role. So why are really haplotypes important? And I have just two examples I want to share here. So this is an example for 2C19. And so one of the first variants that was discovered is what we call the STAR2, and it has two SNPs and both impact splicing. They also all have an amino acid change that is not believed to change function. Now, only many, many years later, there was a variant discovered that was named STAR35. So that was named, I think, 4 or 5 years ago, while the STAR2 is known for over 20 years. And interestingly, this only has one of these two sites. Now, everybody who is in the business of pharmacogenetic testing is testing for this one here. So this one is in an exon, this here is in an intron. So it was, I think, a natural choice of genotyping this one to detect the STAR2. However, if you only test for this SNP here and not for the other one, you will not detect that STAR35, which also has no function. And so in a way, you should actually rather test this SNP here, and then you would catch both of them. So this is one example of knowing which SNPs there are. You need to adapt your genotyping in order to capture these. And interestingly, that STAR35 is rarely found in Caucasian individuals. It is found mostly in people of African origin. And so again, if genotype tests are not adapting to test for this, a variant that leads to no function will not be detected, and the disparity here is that more African Americans here are affected than white people when it then comes to the accuracy of testing. The other example is also for 2C19, and here we have a STAR4 allele which has an amino acid change at the translation start codon, which causes no function. And then there's also an upstream SNP, which is impacting gene expression. So of course, if you have increased gene expression by virtue of that SNP, that doesn't really help you if this SNP here makes it non-functional. However, these two SNPs, while more commonly occurring together, can also occur in twins. And so here the phenotype would be slightly different. It might not always be different for phenotype prediction for clinical recommendations, but here the point also is this SNP here in red is relatively rare and often not tested. And so if you would not test this SNP here, but only the SNP in blue, then you would again end up in vastly different phenotype predictions, because that star 17 here is an increased function allele, whereas this here is a non-functional allele. And so not testing this one will really lead to wrong phenotype predictions. So we are now moving on just to give you some insights into PharmVar. So again, are there any questions so far? Well, let's have a look at PharmVar then. So this here is our landing page. So when you go there, this is what you get. So you still have the opportunity to go to the archive, which essentially takes you back to the old nomenclature page. So you can always track back and see what was the state when we took over. You can go to About, which gives you information about PharmVar, who is on the steering committee. There's also a slideshow that runs you through it. If you are interested in submitting to us, you go to the submission tabs. Here under members is information about who is a PharmVar member. So you see the membership directory. Under submissions, you also see the people who serve on the different gene expert panels. So these are the people who review submissions. So everything is transparent, so you know who will make these decisions. It is not me alone. It is really a committee by majority, which I think is really important. Here under resources, you find a list of publications. You also find information about our API services to pull data down, of course, contact information. And most important is probably the genes tab here, because this is where you get to the gene pages. And so this is essentially what it looks like when you get there. And I have highlighted here important features I would like to point out. So in our header here, we first have, gene names, links to external resources where you can download gene information. Then what is also really important, and a lot of people overlook this, are these documents here. So in these documents, there is important gene information. So what reference sequences are we using? Why? What is the difference between these? Is there anything else you need to know? There are also gene-specific examples of what we display on the page. So this is really important information in the most comprehensive way possible to give you enough insights to really use that information to the maximum. In the other document, here we keep track of important changes. So if you wonder why, something might have changed, why might an allele have disappeared, for example, this is where you can find information. It also lists new submissions, who has submitted, what allele numbers they were given. So there is an entire log of alleles we have defined, which can be very helpful. And in the third document, this is an extensive document detailing structural variation. And so this is now, I don't know, almost 15 pages long for 2D6, because there is so much for these duplications to detail and people to know. Then in here, you can now choose to really see the variance in respect to different reference sequences. So the LRG is a fixed sequence that will no longer change in the future, and this corresponds 100% to the current reference sequence issued by the NCBI. And we have chosen to use the LRG because, as I said, this sequence is locked in, it will no longer change. Whereas reference sequences, for one reason or another, are not stable, so there might be changes the NCBI is making in the future. You can toggle over and see then these positions on the transcript. You can also go over and see what is the chromosomal position on genome build 37 or 38. And we also have here for 2D6 a legacy reference sequence, which was extensively used in the past, but differs in a number of nucleotides within the gene compared to current ref sequences. But it makes it easy to cross-reference positions that have been used in publications in prior years. Then you also do have different filter options, custom views. You can sort for a number of qualifiers here. You also have the choice of 2 different count modes. So you can really display the position counting from the sequence start or from the ATT start codon, which can be helpful because different gene communities might just use different count modes for publications. So that is very helpful and really took the pain out of a lot of calculating the same SNP on different sequences and count modes back and forth, which of course is very error prone. And then we also, here is a graphical view, which I will show you on the next slide, because these gene pages, depending on which gene, are quite long and difficult to see. And you might really just have interest in comparing selected alleles with each other. So this is where this graphical display tool is really helpful. Then you have here selected references, typically the ones where the allele was first described, and whoever has deposited information for the same allele at a later time. Here, what we call haplotype evidence levels. So there are 3 levels: limited, moderate, and definitive. And they really describe on how much evidence do we have that a particular haplotype exists. And so a number of those do have definitive level. So going back into the literature, but also using more recent data, we are sure that this haplotype or all these SNPs that are listed are existing in this form. Some other haplotypes where there is limited evidence, these might be based on just partial information. So for 2D6, there is no information regarding SNPs and introns, no information regarding SNPs in upstream regions. And so we are still waiting for these particular alleles to be discovered in new individuals so that we can submit these again to PharmVar to say, well, this is a haplotype we need to revise, or this is a haplotype which does not change, and then raise the evidence level. And we also have introduced what we call core allele definitions. And this is essentially what you could also call a main allele. And then we have listed under the core definition a number of suballeles. And so all the suballeles, what they share in this example shown here are these 2 amino acid changes. But each of these suballeles have additional SNPs. They could be in the upstream region, there could be synonymous SNPs. And just to collapse all these suballeles into that core allele, we have developed a set of rules which we follow to essentially collapse or expand into these suballeles. And all alleles listed under one allele name are, to the best of our knowledge, having equivalent function. So all STAR2 alleles have been categorized by the CP group as normal clinical function. So this is in essence what you find on the gene page, and then of course there are also link outs. As you click on a SNP, a variation window slides in from the right, where you again see all these different positions side by side. There is a link out to dbSNP. You see the RS number there, and you also have some limited information regarding the SNP frequency. So it's not allele frequency, but the frequency of the SNP, which you find there. And by clicking here on the allele name, you will also find more information here. And lastly, each haplotype also has a unique database identification number, which we call the PV ID number, or the PharmVar ID number here. Okay, so this is what the comparative allele viewer looks like. So you have essentially a dial pad where you can just click the alleles which you want to compare. You hit the compare button here, and then you get a graphical comparison which looks like this. And here you can see very nicely that, for example, that SNP here in this yellow box, you see this SNP occurs in all these alleles where it's in blue, and it's in gray, indicating that this SNP can occur in some suballeles of STAR4. So it's not always present, but it can occur in some alleles. And so, in other words, if you just test for this SNP, you could have a STAR4, a STAR10, and so on and so forth. So there's another SNP here, which also occurs in all these alleles, often even more than what I have picked here. And so if you want to really distinguish these alleles, you would need to do more testing. You would then need to test for SNPs that are unique to some of these. And all SNPs that are unique to one allele have that little PharmVar logo. So you see this SNP here is unique for STAR100. This SNP here is unique for STAR4. But none of the other SNPs here are unique to just one allele. So it's always kind of a process of exclusion. So the more you test, you are then excluding that you have a STAR49, or you exclude that you have a STAR114. And only in principle, if you have excluded all of these, you can then really default it to a STAR10. If you don't do additional testing, you could theoretically have a STAR10, 36, 49, 100, 114, and they are functionally not necessarily equivalent. So again, you will have different phenotype predictions based on the amount of genotyping you have done. And that is really the challenge and the limitations if you do panel-based genotyping. If you only pick the most common one and only test for these, then for some alleles, you cannot say for sure whether they are there or not. I mean, first of all, what you don't test, you don't see. But there's also this defaulting process where you might determine you have a STAR10 because this SNP is present, but if you have not excluded that you have a 49 or 100 or 114, these might default to a STAR10. So, okay, this was a lot to take in, I think. So I just take a breather and give you the chance to ask any questions for that. Anything? Nothing? Are you all thinking? Probably. Okay, so as I have mentioned before, PharmVar is also expanding to include important non-cytochrome P450 pharmacogenes. So after all, we have chosen our name carefully. We want to cover pharmacogenes of interest, not just cytochrome P450s. And so the first gene that is not a P450 is NUT15. We have launched this now 2 years ago, and that is important along TPMT for cancer treatments. For example, Allele, which is really a very common cancer childhood disease. We have launched DPYD, which is also a gene that is involved with certain cancer medications. Now for this one, we have developed a different gene page format. You can look up if you like to. And we are starting on the first transporter, SLC01B1, which has been implicated in statin metabolism. And so we are just starting this here. We have also a publication that describes the process we went through as we developed this gene page. And I just threw in here a slide to make you aware of all our publications. So this is the first one where we describe the transition to PharmVar, and then the evolution of PharmVar describes certain features we have implemented over time. Then in that third one, we describe how we are working together with other resources, and that was one of the papers you looked at yesterday. And we have also started a series of review papers, which we call the PharmVar gene focus papers. So our first one was on CYP2D6. We had another one half a year ago on 2C19. There is a third one under review, which is on CYP2B6. And a fourth one is right now being written, which is on 2C9. So these gene review papers really give you the breadth of what to know about the gene, but also what we do with PharmVar, and highlight different challenges or features, things that are unique for this gene. And I also want to highlight that we have developed templates for authors to describe genotyping methods, also how to report these alleles or diplotypes and how they are translated into phenotypes in a more structured and unified way, because reporting of these pieces is sometimes all over the place, or authors refer to other publications, so you have to backtrack over a lot of papers to even find out what have they done. And this makes it really difficult for database curators to get the information if it is not provided in a very structured way. And these templates, I think, will be very helpful for people just to report these findings the same way each time. It also helps them out because they essentially just have to fill a table that is already there. So these templates are available as supplemental materials to these gene-focused papers. And it will, of course, be your decision whether you want to put these tables in the supplement of your papers or make it a main table in one of your publications. Okay, now from here on, I just want to briefly go through different applications of where PharmVar really plays an important role. So this overview was taken from our landscape paper, so you probably have already looked at this in more detail. So first of all, the PharmGKB knowledge base is really the go-to knowledge base for pharmacogenetics and pharmacogenomics. And as you can see here, they have annotated a large number of drugs they have these beautiful curated pathways. They work closely with the CPIC group, so there are all these clinical guideline annotations and also drug label annotations. They also do have really nice introductions into what is pharmacogenetics and pharmacogenomics, and provide information of what this site provides. So I really encourage you to take advantage of these introductory information. And I also want to point you towards these gene-specific information tables. So when you go to their webpage and just scroll down, you find these. And here you find for all the genes for which there is a CPIC guideline, you find these reference tables where again you find information for allele definition. How are they translated into phenotype? How do, or what is the function for each allele? How is a diplotype translated into a genotype? And so on and so forth. All that information is here on the PharmGKB, among much more. And these beautiful pathways, you might have seen them. These are also made by PharmGKB. There are plenty of those. They're all free for you to use. Just please cite where you get them from. So these are really widely recognized. Now the CPIC, the Clinical Pharmacogenetics Implementation Consortium, so this is really housed not far from where you guys all are. So this is, they are at St. Jude's. And so, of course, as they are using also or make these recommendations based on genotype, they rely on genetic variation in a very structured way. And so what is this consortium in the first place? So they started out now about 12 years ago. They were formed officially in 2009, and really their focus is to overcome the barrier to clinical implementation of pharmacogenomics, because there's often not really information freely available that tells physician of what to do with a genotype. And so to provide professionals, prescribers, the information of what to do with a genotype, if that information is available, they have decided to develop these guidelines so they can use these guidelines to optimize drug therapy. The CPIC, however, is not making recommendations of whether you should test. And this has liability issues. I mean, that's another completely other issue, to tell physicians whether they should test, but to give them guidelines and recommendations of how to interpret that genotype if they decided to order it. And these guidelines are peer-reviewed and published in partnership with the CPT journal. They work very closely together with the PharmGKB, where you also find all the information, data, and updates. So all the guideline information you can find on the CPIC website itself, but it is also fully incorporated into the PharmGKB. And anyone who has interest in pharmacogenetics can apply for membership and be on their monthly calls to really have all these updates of what is going on. You can have the opportunity to comment to guidelines that are being developed and ready for submission, provide feedback, or even participate as an author should you have the experience to work with them on that level. And so there are too many guidelines to list, but I have compiled here the ones out for 2D6. And again, this gene, because it metabolizes so many different drugs. There are 6 guidelines right now, and you can also see that guidelines are periodically updated. So the one on codeine first came out in 2012. It was updated in 2014, and now it is under review after this guideline was expanded beyond codeine. There are other opioids and other genes that have been incorporated in this guideline. That should hopefully be published fairly soon. We hope towards the end of the year, and if not, very early next year. This one here on antidepressants was also updated in 2016. This year on the SSRIs is currently being updated. And then there's also one on ondansetron and topicitrone. So these are anti-nausea drugs. There is one for tamoxifen, which is a drug that is used to treat breast cancer. And we have one for atomoxetine, which is often given to treat ADHD. So many of these are very popular and frequently used drugs, and this is why there are guidelines out there. And in these tables, you can see this is the heart of a CPIC guideline. The recommendation is driven off the phenotype, but the phenotype essentially is the product of a diplotype or the genotype. And you can see here again these star numbers, and all these allelic variations are sourced from PharmVar. So this is the source where these allele definitions come from. And because the CPIC is making clinical recommendations, we are very aware that we need to set the bar high for these haplotype definitions. And really also these evidence levels can help us with this entire process of the interpretation of these different haplotypes and diplotypes into phenotypes. This slide is really here to remind me to let you know that these guidelines, there are short videos available on YouTube which you can look at. So that's the easy way to familiarize yourself with a guideline. So please go there and check them out to see what these guidelines provide. Now, when it comes to clinical test reports, these are also using STAR nomenclature. And of course, as testing companies use the STAR nomenclature, there needs to be a source, a resource where you can go and look up, well, what do they mean? And this again is coming back to PharmVar. Now, whether you should test and where you should get your test, which company offers a good versus not so good test can be quite daunting. And so I cannot go into this really extensive diagram here, resource to get you on the way to make decisions of whether testing would be recommended, could be useful, and also gives you quite a bit of ideas of what you should look for in a test. Then along the same lines is really to sort out what is a good test and what is not a good test and what should really be tested. And so this question was taken up by the Association of Molecular Pathology, or AMP, and they have developed a two-tiered system of alleles they recommend a test should have. So the tier 1 alleles are the minimum panels of variant alleles that should be tested, and tier 2 are those which gives you an extended panel of variants to be tested. And again, these variants are based on the PharmVar allele definitions. And so they have 3 papers out there. 2 are shown here. So they give recommendations for 2C9 and 2C19, the tier 1 and tier 2 alleles are shown here. The third paper provides recommendations, particularly for warfarin testing, which alleles should be tested for that. And we are wrapping up right now a recommendation for 2D6. So again, that new paper will show which are the alleles that at a minimum should be tested, and gives also more extended alleles that a test should encompass. And so that makes it much easier to compare what a given test, do they follow the tier 1 or tier 2 recommendation? So that gives you some sense of how good a test is if you have these recommendations here at hand. But then again, it's a free country here, and so companies, of course, offer different tests. They want to distinguish themselves, and so it can make it really hard for you to sort out, is the test really good or not? And these papers are very helpful in giving you information to check what companies offer. Then there is another group we are working with, which is the Genetic Testing Reference Material Coordination Program. So of course, anybody who is doing genetic testing, you always want to have positive controls. So you should have ideally positive controls or controls of different genotypes, which you can run along. And so this group has taken up the task to work with different academic laboratories to test Coriell samples, which are the reference materials which are widely available, and run them through different platforms and testing and provide that information. And so you can then see if you test certain DNAs with different kind of tests, what is the genotype you get if you do if you run it through these different platforms. And so this is also very helpful resource how you can get positive or negative control samples for certain genotype testing. And we had now almost a year ago an extensive paper on just CYP2D6, where we were able to add quite a few interesting samples with very rare and unique genotypes to that panel of control samples. And we are currently working on an expanded testing for CYP2C9, 19, and 2C8 using next generation sequencing data and different bioinformatic haplotype calling tools to see how good these tools are, what are we missing, what are the weak points, so that we can identify novel alleles, then bring them, submit them to PharmVar, define them to give these tools then more variants to incorporate and hopefully make the calling more accurate. And this is essentially just summarizing tools and algorithms for genotype data interpretation. So as you can imagine, especially if you have next generation sequencing data, you can possibly not call all these different diplotypes by hand anymore. So you need good tools that allow you to interpret these data. And so certain commercial genotype platforms, they provide their proprietary algorithms to do so. And then there are also freely available tools, which I have listed here, which all use the input from PharmVar. So these are the star alleles they are going to call. And then they do their kind of, magic, their algorithms. They all have different ways to come up with these haplotypes and then spit out a genotype at the end. And hopefully that genotype is the correct one. And there is still very little information in terms of how good are these tools, especially when it comes to rare alleles, because there are just not many out there. then of course we still have novel alleles, and no tool can call what's not known. And so going through these exercises and still discover novel variants and also better define existing variants and then feed that information back into PharmVar, and therefore allowing these tools then to update the allele definitions is really helping to make the accuracy of these tools much better. So we have developed Allele at my institution, and we have gone really through having a lot of ambiguous calls. And as we were going back and looking at these haplotypes, defined these haplotypes better or added new alleles, then came back, reran everything, then we were able to come to the correct call. So we are still going through circles of adding new alleles, feeding those back into these tools to improve their call accuracy. So that is still quite a bit of work I think we need to make here before we can reliably use these tools for next generation sequencing. Unless, of course, we are filtering out just a subset of these SNPs or alleles where we are much more confident calling. And then there is one more resource I want to just introduce. This is the Pharmacogenomics Clinical Annotation Tool, or PharmCat, which is developed together with the PharmGKB. And what they want to do is build one pipeline that really goes from the sequence to the processing to the allele calling, and then all the way down here to a final report that includes all these CPIC guideline recommendations. So really provide sort of a one-stop, one-shop kind of pipeline going from the data, which can be sequencing or panel-based, but this platform will essentially only look for variants which have been curated and are incorporated into these guidelines. So it's not looking for anything new, but only for those that are already known and which can be interpreted. But this is also really great to have that one platform and one pipeline to incorporate all this. And as I said, PharmVar again is a part of really contributing to that in terms of the genetic variation. And this is my last slide here. And so as I mentioned in the beginning, pharmacogenetics is really just one piece of the puzzle. There is so many different kind of pieces that need to be in place, which is not just all these different resources, but also basic research, medical research, precision medicine. Only if we really work together here is this piece coming together, and hopefully pharmacogenetic will find a place in the future and is more widespread in its application. And with that, I will take any questions.
So I think we went a little bit over on time here, but maybe we can ask, maybe we have like one or two questions and then move more to like an informal Q&A session.
Hey, hi. Can I ask you a question?
Sure.
All right. Yeah, this has been a great talk. Thank you so much. I just wanted to ask if these star allele names are permanent, and if not, how does the renaming of these names affect things if more information about allele is known over a period of time?
So a star allele is not necessarily fixed. So if there is new information for an allele, so let's say there is one with a low evidence level, and now there is new information, that tells us there are additional SNPs on that haplotype, the star number will stay the same. The PVID number, however, changes because the PVID number is unique to a haplotype, whether it's the right one or not. So a PVID number only links to one particular haplotype. So in that case, the star number remains the same. However, if there is an allele that is right now a suballele of a star 1, it has, for example, a SNP in the upstream region. And now new evidence tells us that this upstream SNP is in a regulatory element and changes gene expression. And if there is enough evidence that shows that this allele, because of that upstream SNP, now has increased expression, therefore leads to increased activity, then we will pull it out from under the star 1 category and give it its own star number, because now it is functionally different than all the other star 1 alleles. So with that, the star number will change. Okay. However, the PVID number will, in that case, stay the same because we are not revising the SNPs on this haplotype, but we are pulling it out because new information became available.
Okay, but if the functional interpretation changes, then that would change the ID, the PVID?
The PVID number is really just telling you what is the haplotype. So if the haplotype itself does not change, the PVID number will not change, but the star number may change. Because all alleles that are captured under one particular star number are functionally, or should be functionally, the same based on current knowledge. But new knowledge might require us to pull out a subvariant, for example, and give it a new star number.
You do that for copy number variants, Andrea?
The copy number variants, they are not listed in the database right now because these are really a different beast. And so these are for 2D6 listed in the PharmVar, and I just pull that over here. So if you go to genes to 2D6, which is here, to the structural variation document, you see here there is a 15-page document really telling you everything you need to know about structural variation in 2D6. And these different structures are really very difficult to keep track in the gene page, where we really list each and every variant and report it out compared to a reference sequence. And doing that for these structural variants is very complicated. And this is why we have developed this document. And this is right now provided as a PDF page. And we hope to switch these over sometime in the future also to HTML sites so that they become better searchable. But as so many things these days, we are limited by funding. So if we had more funding and could hire more of a web developer, things would go a little faster. But for now, this is really a 15-page long document, and you can see there is a lot of tables. And because there are so many different of these variants, it requires a lot of, slicing and dicing, and which is similar and which is different in terms of function and so on and so forth.
Thank you.
Rob, does this seem like something that could be addressed with some of the pangenome graph-based tools?
Yeah, I think it certainly could. The CNVs have been detectable with the Roche chip for a long time now, but Andrea would know how accurately they're diagnosed with the Roche and other chips.
I'm not exactly sure what you mean with the Roche chip. Is that the DMET chip? Or, there are so many different platforms out there. And of course, for copy number, you need to dive really into the weeds of a test to know what exactly is a test testing, and then to what kind of copy number variants would be identified by this testing. And again, it's probably not a straightforward answer.
Okay, so I'm going to go ahead and stop the recording, and we'll go continue this with more of an informal Q&A session. But I would like to thank you. That was a very enlightening presentation, and there's a lot of tools available and a lot of work that's been put into this. So thank you for sharing that with us today.
You're very welcome. Thank you.
Thank you so much.